Hello and welcome to another episode of Research Talk. Today we are pleased to have Mr. Bharat Singh Patel with us. He's from Prayagraj, Uttar Pradesh. Bharat completed his BSc from Ewing Christian College, Prayagraj, and went on to earn his MSc from IIT Jodhpur. He's currently a research scholar at the Indian Institute of Science, Bangalore. His research interests include design, synthesis, and biophysical analysis of glycoconjugates. In today's talk, he'll be presenting his work, which entitled as Multivalent Chitobiose Self-Assembled Glycostructures as Ligands to Lysozyme. So Bharat, welcome to the talk and the screen is yours.
Thank you, Mahesh, for kind introduction. As we know, lysozyme is well-known antimicrobial enzyme that naturally recognize chitin derivative oligosaccharide such as N-acetyl glucosamine units. However, individual carbohydrate-protein interactions are weak and transient. The central question addressed in this work is whether multivalent presentation of chitobiose on self-assembled supramolecular architecture can significantly enhance lysozyme binding and modulate its enzymatic activity. This study integrates concepts from glycochemistry, supramolecular self-assembly, and biophysical characterization to demonstrate how weak monovalent interaction can be transformed into strong and functional multivalent interactions. This presentation, I will first introduce lysozyme and its biological function and the pathological relevance. I will then discuss chitoolgosaccharides as natural ligand for lysozyme and the limitation of the monovalent interactions. And then I will introduce the concept of multivalency and describe the design and synthesis of the chitobiose-based glycolipid. Finally, I will present a comprehensive biophysical and biochemical evolution of glycostructure lysozyme interactions and their effect on antimicrobial activity. Let's begin with the introduction. Lysozyme are naturally occurring enzyme found in wide range of organisms, including bacteria, fungi, plants, and animals. In mammals, lysozyme is present in secretions such as tears, saliva, milk, and serum, where it plays a critical role in innate immunity. Lysozymes are classified in three major families: chicken type, C type, invertebrate I type, and goose type Z type. Among these, C-type lysozyme consists of 129 amino acids with a molecular weight of approximately 14 kDa, is most extensively studied and is used as a model enzyme. In this work, lysozyme catalyze the hydrolysis of beta-1,4 glycosidic linkage between N-acetylmuramic acid and N-acetyl glucosamine in bacterial peptidoglycan, leading to bacterial cell lysis, in which the glutamic acid and the aspartic acid of the enzyme taking part in the general acid catalyzed mechanism pathway. It shows lysozyme shows several applications in medical, like treating infectious disease, wound healing, antibacterial, antiviral, many more. Even though having such medical application, but excess secretion of the lysozyme leads to the nephropathy, neuropathic pain, and several other diseases. Therefore, the ability of modulate lysozyme activity in controlled reversible manner is of considerable biomedical interest. Chitoolgosaccharides are low molecular weight derivative of the chitosan polymer and exhibit excellent water solubility. They possess a wide range of biological activities, including antimicrobial, antioxidant, and anti-tumor effect. Importantly, chitoolgosaccharides are natural ligands for the lysozyme because they mimic the enzyme native substrate. However, when chitoolgosaccharides bind individually to the lysozyme, the interaction is very weak, typically characterized by dissociation constant in the millimolar range, which limit their effectiveness as inhibitor or the regulator. As we can see here, several glycosidase inhibitor has been reported in the literature, many of which are carbohydrate based. Despite of structural, most of these inhibitors rely on the monovalent interactions and therefore suffer from low binding affinity. This highlights a fundamental challenge in glycobiology, achieving strong and selective binding using carbohydrate. This limitation motivates the exploration of multivalent strategy. Carbohydrate protein interactions are governed by hydrogen bonding, electrostatic interactions, metal coordination, water-mediated bridges, and the hydrophobic interactions. Each these interactions are weak. As a result, the single carbohydrate protein interactions is weak in nature, and the dissociation constant lies in the millimolar range. So the question, how one can increase the carbohydrate protein interactions affinity? The answer is the multivalent effect, where the logarithmic enhancement of the binding affinities of the cluster sugar ligand with the linear increase of the sugar residue. Where this phenomena arise from the cooperative effect, reduce dissociation probability and statistical rebinding. Multivalency is widely exploited in the nature, particularly in the glycan-mediated recognition process. So our primary objective in this study was to design and synthesize self-assemble chitobiose containing glycolipid, where we can tune the surface density of the glycolipid. We aim to systematically evaluate how multivalency affect the lysozyme binding affinity, protein conformation, and antimicrobial activity. The ultimate goal was to demonstrate control modulation of the enzyme function rather than the irreversible inhibition. For the synthesis of the glycolipid, we had taken the protected disaccharide thioglycoside and the diethylene glycol linked stearic acid. We activated this donor by using NIS timestiflate in dichloromethane at -18 degrees centigrade for 1 hour. We got the protected glycolipid that upon using hydrogenation condition leading to the removal of this benzyl group, as well as the azide to amine conversion, followed by this amine acylation using acetic anhydride in pyridine, we achieved glycolipid. Since in this glycolipid, so there is the ester bond, and since it contains this hydrophobic part as well as this hydrophilic part, so it will undergo for self-assembly. The critical aggregation concentration determination, we have taken the 1,6-diphenyl hexatriene as a DPS probe. So DPS in aqueous medium, it will not show the fluorescence, but once it will intercalate inside the hydrophobic region, it will show the fluorescence. CSC values are essential because multivalent presentation of the ligand occurs only above this concentration. All subsequent interactions studies were performed at the concentration above the CSC value. So as we increase the concentration of the glycolipid from 10% to 100%, the CSC value also come down 25 micromolar to 5 micromolar. So by seeing this CSV value, we can say like above this value, like 25 micromolar, there will be self-assembly. So all the experiment we have conducted above the CSV value, somewhere 100 to 200 micromolar concentration. So first, we wanted to see the solution behavior of the glycostructure. For that, we undertook dynamic light scattering measurement to determine the hydrodynamic diameter of the self-assembled glycostructure. In the solution, the observed size distribution were consistent with the vascular assembly and indicating good colloidal stability. These results confirm that the glycolipid form well-defined nanostructure suitable for the protein interactions. From 10% glycolipid, the Z value was 184 nanometer. As we increasing the concentration 10% to 100%, the Z value increased to 77 nanometer. For this experiment, we have taken the concentration of the glycolipid 200 micromolar in the sodium acetate buffer at pH 5. We have taken here sodium acetate buffer at pH 5 because the most stable and the most active form of the lysozyme present at pH 5 in the sodium acetate. Then we wanted to see the surface morphology of the glycovesicle. For that, we undertook atomic force microscopy and the scanning electron microscopy. Through that, we can direct visualize the morphology of the glycostructure. Both techniques reveal the vesicle-like structure supporting the DLS data. This consistency between the solution-phased and the surface-phased characterization technique strengthens the reliability of the structure conclusions. As we increase the concentration of 10% to 100%, the size of vesicle also increased. This is the same for AFM and the SCM images. Since our glycolipid contains ester moiety, so that will not survive at the acidic and the basic pH. For that, we wanted to see the time and the pH dependent stability of the glycovesicle. For that, we undertook the emission spectra, where we have taken the DPH as a probe, and then we added our we have taken different pH of the solution. What we found is at the neutral pH, the stability of glycovesicles were more compared to the acidic and the basic pH. And also from 10% to 100%, we can see the entrapment of the DPS was more compared to the 10% and the 50%. Further, we have taken the DLS analysis and the AFM analysis for the lysozyme glycostructure complexation for all the formulated 10%, 50%, and 100% glycolipid. So after addition of the 10% of glycolipid into lysozyme, so as time increasing, the diameter increasing, that suggests there is a interaction between the lysozyme and the glycostructure. And as we can see in the AFM also, the size of the glycovesicle becoming the diffuse. As we increasing the concentration of the glycovesicle 10% to 100%, the size of glycovesicle increased to 510 nanometer. After 10 hours, it become 2106 nanometer. And in AFM, we can see is completely diffuse in structure. That shows that there is a progressive size increase, reflect multivalent cross-linking between the lysozyme molecules and the chitobiose decorated self-assembled structure. This behavior is indicative the controlled supramolecular complexation rather than the non-specific aggregation. Here we have taken the lysozyme concentration 25 micromolar and the glycovesicle concentration was 200 micromolar. And further, we have taken the fluorescence spectroscopy for the lysozyme glycostructure complexation study. Intrinsic tryptophan fluorescence of the lysozyme decreased upon interaction with the glycostructure. This quenching suggests that change in the local environment of the aromatic residue that is responsible for the fluorescence, confirming close proximity between the protein and the multivalent chitobiose ligand. As we can see in this data, like for 10%, there is a like more decrease in the intensity compared to the 50 and 100% of the glycovesical structure. Further, we can see in the UV experiment, there is also change in the absorption intensity, further support the DLS and this fluorescence spectra. There is a like lysozyme glycostructure complexation. Because of that, there is a decrease in the intensity. Further, we have taken the CD spectroscopy for the lysozyme and glycostructure complexation. CD spectroscopy revealed that the characterization alpha has helical secondary structure of the lysozyme remains largely unchanged upon the complexation. This result demonstrated that multivalent binding does not denature the protein, indicating that inhibition occurs through non-destructive reversible interactions. Further, we have taken the surface plasmon resonance technique to see the glycovesicle and lysozyme complexation. So first, where it employed the quantitative analysis binding kinetics. So we have taken the CM5 sensor chip where we have immobilized lysozyme by using sodium acetate buffer, and then we pass our glycovesicle above the CMC value at the concentration varying 20 to 50 micromolar at the flow rate of 30 microliter per minute. So as we can see in 10% of glycolipid lipid vesicle, there is a, as the concentration of the glycostructure increasing, there is a increase in the response unit. As we increasing the concentration of the glycovesicle, so here we can see there is a drastic increase in the response unit. And this sensogram showed increasing response unit with the increasing chitobiose density, indicating stronger binding as the concentration of the glycovesicle increasing. This direct correlation between ligand density and the binding response provide strong evidence for the multivalent enhancement. So here we can see with respect to all the concentration of the glycovesicle for 10%, 50%, 100%, K and KD value and the binding and affinity. This was analyzed by 1:1 Langmuir model, revealed that in case of 50% glycolipid and lipid composition exhibited the highest association constant compared to the native chitobiose lysozyme complexation, that is 3.5 into 10 power 3 per mole. There is an increase of 2 to 4 order of magnitude of the glycostructure interaction with the lysozyme. This clearly demonstrates the power of the multivalency. Further, we have taken the lytic activity of the lysozyme on the E. coli. The functional consequence of the binding was evaluated using lysozyme-mediated lysis assay against E. coli. The presence of glycostructure, if you can see here, this black one is the bacterial cell with the lysozyme. There is a decrease in the optical density, but once we added our glycostructure to the lysozyme, that delayed the optical density, that slow down the activity of the lysozyme. This confirmed that there is a strong binding translate into the functional modulation. This delayed lytic activity suggests the glycostructure regulate the lysozyme activity rather than permanently inactivate the enzyme. The controlled inhibition in this particularly important for the therapeutic application where the reversibility is desired. In conclusion, we successfully synthesized, designed multivalent self-assembled chitobiose containing glycostructure with a tunable ligand density. This system exhibits dramatically enhanced binding affinity towards the lysozyme while preserving protein structure and the enable control modulation of the antimicrobial activity. This work highlights the multivalency as the powerful strategy in the carbohydrate protein interactions. This work has been published in the Bioorganic Chemistry. For further details, you can scan this QR and go through the paper. Thank you, Turning Point.
Nice work and nice presentation, Mr. Bharat. If you enjoyed this session, don't forget to like, share and subscribe. We'll meet you in the next episode, next session. Thanks for watching.